Thank you for inviting me, Yelena. This is my disclosure. And so today I'm going to tell you about, I've trained at Sloan Kettering, and I work primarily across the street at Rockefeller, and I do a small amount of clinical work, mainly to to learn from my clinical colleagues. I'm going to tell you a story today about how we started probably 8 years ago or something in the lab by asking a simple question of how colon cancers become more metastatic and more aggressive. And we were able to identify a key gene involved in metastatic progression. And then at that same time, we began long-standing collaborations with a number of clinicians here, Pat Patey, a surgeon, Yelena, Len Saltz, Peter Kingham, Elisa Distancina et al. And this allowed us to to do clinical correlative studies and to really focus our attention on more clinically meaningful biology to pursue. And that's been really remarkable. This focused our attention on a gene, and we went on to define the pathway and its biology. And then I'm going to tell you a bit about how we've gone on to move that towards a small molecule experimental therapeutic that targets a metabolic pathway that we think is important in colon and gastric progression. And so this is what we started with. We were interested in liver metastatic colonization. And you guys are all aware of this. And what we know experimentally and this is something I didn't know a decade ago, but this is a really inefficient process. So experimental studies have shown that 1 out of 10,000 or 1 out of 1,000 to 10,000 cells, if you inject them into the circulation, a portal circulation, only one of those cells will actually be able to colonize the end organ. So this is a major bottleneck process, and I know just in the last talk we're talking about importance of adjuvant therapy and prevention of the process. I'm hopeful because of this really inefficiency of the process and the fact that it's a bottleneck process. But why is it a bottleneck process? Well, cancers of the gastrointestinal tract, these cells are exposed to a hypoxic environment, both in the circulation, sort of the portal circulation, for example, but also as they arrive in the liver where regions of the liver can be hypoxic, but also colon cancer or gastric cancer cells metastasizing to this organ have to compete with hepatocytes, which are professional importers of glucose. So these cells are under intense metabolic stress, both hypoxic stress and glycemic stress. And that's not optimal conditions for growth or survival. So many years ago, Xiaomin Liu, a graduate student in the lab, I think about 8 years ago, became interested in this problem, and we didn't know where to start. And so we're sort of boneheaded, and we just say, look, we don't know where to start. Let's just get human cells, get cancer cells from a patient, introduce these cells into the livers of the mice, label them with a luciferase gene, introduce 2 million cells, and then wait for 3 or 4 months and see if tumors grow. When the tumors grow, we take those tumors out and we inject it into another mouse. And we keep doing that until after a couple generations, we start seeing that these mice get ill now after 3 weeks instead of 4 months. We take those nodules out, and those are more metastatic subpopulation that we've enriched for. We've sort of purified gold without any markers because we don't really know what markers to look for. And so does this actually work? So it does. So if you take the parental colon cancer cells, which are not that metastatic, each dot represents a mouse. This is the photon flux, the luciferase signal on a log plot. And you can see that the poorly metastatic cells make this tiny little speck. That's the liver ex vivo. But if you introduce these selected highly metastatic sublines, you can see 100-fold, 80 to 100-fold enhancement in their ability to form metastatic colonies, okay? So now we have the cells all from the same patient. We're comparing apples and apples. They're different. These two sublines, their third-generation derivatives, are different from the parental cells simply in their capacity to form these colonies. And so then what we did is, about a decade ago, we showed that one mechanism by which rare cancer cells, breast cancer cells, for example, can form metastases is by upregulating or downregulating specific genes. And they do that by modulating small RNAs, microRNAs. So if it's a gene that allows them to form metastatic disease, what they might do is to shut down a microRNA that inhibits it and thus overexpress that gene. So what Jiamin did was he speculated that there may be some microRNAs that can actually suppress the ability of colon cancer cells to form metastatic disease. And so he looked for them by taking 680 human microRNAs, putting them in viruses, introducing them into a population of colon cancer cells, injecting these colon cancer cells into mice, getting nodules, taking those nodules out and looking at each of the cells and asking, are there any microRNAs that aren't present anymore? Because when they were present in that colon cancer cell, it inhibited the ability of the colon cancer cell to form a liver metastatic colony. Those cells would die and drop out, and then we could quantify that. And we did. And Jiamin was able to find two microRNAs, two small RNAs, that when he put into these colon cancer cells, they inhibited the ability of the cells to form metastatic disease. So now I can show you that data. Here are the highly metastatic colon cancer cells. We find that they shut down these two microRNAs, and they can form metastatic disease. Now, if you re-express these microRNAs that he identified, 483 or 551A, you express them again in these cells. Now you can see a very massive suppression of metastatic colony formation. You can do the converse experiment. Take colon cancer cells that are poorly metastatic and inhibit either microRNA, and you can see a tenfold enhancement of their ability to form metastatic colonies. Each dot represents an individual mouse, so there are a lot of mice here. And so, in collaboration with Pat Patey and Xiaoxi Zhang in his lab, Pat had over the years collected many primary tumors and liver mets from patients, and we analyzed over 60 of them, and we found that the liver mets had lower expression of each of these microRNAs than the primary tumors. And that was cool because we speculated that these microRNAs suppress this process. So it would make sense that during colon cancer metastatic evolution that they would shut down these two microRNAs. So what's the mechanism? So we couldn't figure it out. It took us a year, but what we did was to simply say, let's look at the cells. Let's watch this process. So what we did was to take colon cancer cells, label normal cells in green. You can see these green cells in the liver of the mice. We're imaging it with multiphoton microscopy in the liver. And then the red cells are cells where we've inhibited each of these microRNAs, okay? When we inhibit these microRNAs, we get more metastases, okay? Now you can see at day 0, it's 50-50. You see half the cells are green, half the cells are red. But you see by day 3, the green cells are gone. They're the ones that are expressing the microRNA. But the red cells, which are inhibited for the microRNA, they're present, and they're actually growing. And the same thing for this microRNA. And we know that the reason the green cells disappear is because they're dying. They're undergoing apoptosis. So the presence of each of these microRNAs causes these cells to die as they try to colonize the liver. So what's the mechanism? So microRNAs negatively regulate sets of genes. So what we did was we put these microRNAs in the metastatic colon cancer cells, and we looked for what genes became downregulated. And what we were surprised to find is that while each microRNA downregulated, let's say, 10 or 20 genes, they shared a common gene. They shared one gene in common. They convergently targeted a single gene together. So we became interested in that gene because it suggested to us that that may be what's mediating the effects, since both of these microRNAs are negatively regulating it. What is that gene? That gene is creatine kinase brain. It's a gene that's normally expressed in our brains, okay? And what we find is that when we inhibit in the colon cancer cells these microRNAs, now this is protein level, its protein level goes up. It gets turned on. So highly metastatic cells shut down these two microRNAs. That allows them to overexpress a gene normally expressed in your brain. That gene is called creatine kinase brain. What does creatine kinase brain do? I didn't know much about this, even though I look back at my old biochemistry text and they talk about creatine. So creatine, you guys all know about creatine. We check it in our chemistries on our patient. We check renal function for it. It's a very abundant metabolite in our bloodstream, 1%, okay? What this kinase does is it takes a high-energy phosphate and puts it on this molecule right here. Now, that phosphate is 50% more energetic than the gamma phosphate of ATP. This molecule, phosphocreatine, is highly energetic. It's our energy buffer. In our muscle, we have 10 times more of this than we have of ATP. So it's like currency. When you really need ATP, what your body does, it converts this stuff to ATP rapidly and uses it for energy. Got it? So your brain has high abundance of it. Your muscle has high abundance of creatines, different creatine kinases. But the one I'm talking about is the one primarily expressed in the brain. Now, what about this gene? Is it important to metastasis? We find it is. When we knock it down, we get over 100-fold reduction in the ability of these very aggressive colon cancer cells to form metastatic disease. And we see this across cancer types of distinct mutational spectra. How about genetics? Lisa Noble did this experiment. She takes colon cancer cells, injects them into circulation of mice. You get massive liver metastatic disease. If she takes away this gene, normal liver, okay? Very little colonies in the liver. It's not absent. You still have small microscopic colonies, but they're not colonizing the liver. We can look at clinical data sets. So this is Patey's samples. We can see this enzyme's expression goes up. We can look at published microarray data sets. This is log plot. We can see it's significantly overexpressed in liver mets. We made a microarray with pathologists at Cornell. A fellow in my lab made it. We can see that its protein levels go up in liver mets as well. So across independent data sets, this enzyme is overexpressed during colon cancer progression. I'm just going to summarize the model. We did a lot of mechanistic studies. So what we find is that, what does this enzyme do? We find that this enzyme, when the microRNAs are shut down, the cell overexpresses this enzyme. Surprisingly, we find that the cells release this enzyme outside the cell. And what we find is that it takes this metabolite that's highly abundant, creatine, and it uses ATP in the tumor microenvironment that can be up to 700 micromolar in the tumor microenvironment to make phosphocreatine, this metabolite, high-energy metabolite. And it imports this metabolite into the cell, and it uses it to make ATP in order to survive the hypoxic and metabolic stress inherent to the tumor microenvironment or the metastatic microenvironment. You might say, this is ridiculous. Why doesn't the cell just bring in ATP? Well, evolution thought it's a terrible idea to allow cells to bring in ATP because that can lead to transformation. So what the colon cancer cells do is they walk around the process. They overexpress a gene they're not supposed to. That's normally expressed in the brain. And they bring this high-energy metabolite in. So if that's the case, then if we get rid of CKB, we should reduce the levels of this metabolite inside the cells, and that's what we find. When we overexpress CKB, we can see a significant increase in this metabolite in the cells. If we deplete this CKB, we see that the levels of this high-energy metabolite go down inside the cells. CKB should regulate the cellular ATP levels. If this metabolite, high-energy metabolite, regulates ATP levels, that should be the case. That is the case. We can take the colon cancer cells, we knock down CKB, and we see that there's over a 35%, 40% reduction in cellular ATP. That might not seem a lot to you, but that is a massive amount. And that sensitizes the cells to metabolic stress. Now, if we provide this high-energy metabolite outside the cell, it repletes the intracellular ATP levels, okay? Now, sorry, we can't see it. So if we block this import of this high-energy metabolite by blocking the transporter, we should also get reduced metastatic colonization. So the transporter for this metabolite is SLC6A8. It can import creatine and phosphocreatine. When we knock it down in the cells, you can see this is the protein level. We don't delete it, but we're depleting it. You can see a substantial reduction in metastasis formation. So then we thought about therapy, okay? So now we have a pathway. It's a metabolic pathway. It seems like as these colon cancer cells progress, they're dependent on it. and we can inhibit it. We can do molecular biology, genetics. It's important. Can we develop a therapy? So what we did in the lab is to look for small molecules that could inhibit this transporter, and we were able to find a mimetic of this metabolite that could block this transporter, okay? And then what we did many years ago was to found a small biotech, which I put in my disclosure, that was really, pretty much everybody there is either a PhD or an MD, and they're all focused on this approach and another approach. And basically what they did was, it was painful, but because I think this is not a classic target, no one's really pursued this. There are no mutations in this gene. There are amplifications of it, but there aren't any mutations of it. So the idea was to see if we can make an inhibitor. So this inhibitor, this small molecule, what we can do is we can label creatine, deuterated, inject it into mice, then give them this drug, small molecule, and then we can measure in their heart, because heart can also uptake this metabolite, the levels of this metabolite. And we can see that when we give this compound, we can, in a dose-dependent manner, reduce the amount of this creatine into the heart. And here's a knockout mouse. This mouse has no transporter. So you can see this is the maximum we can get, and we're doing pretty good in terms of reducing it. When we give this to mice, what we find is that in tumors, this is a mouse tumor. Red is cleaved caspase. You can see dose-dependent apoptosis in the tumors of these mice. This is work done by Isabel, Celia, Andrew, and Shugaku Takeda. So this is a KRAS mutant mouse tumor. Now you can give this to tumors when they get about 70 millimeters cubed, and you can start treating them. You can get tumor growth reduction. This is the KRAS wild-type tumor, 2 different murine tumors. Now, this is another model that responds to this small molecule, not as good as others. This model responds well to 5-FU. We can see additivity with 5-FU, so this is control tumors. When we give the drug, we get tumor growth repression, inhibition, 5-FU, and the purple is the combo. And these are the survival curves. Now here, orange is the combo. So there seems to be additivity with 5-FU. This is a KRAS mutant human cell line now. And you can see when we start treating, this is an athymic mouse. This is an immune deficient mouse. And you can see that we can start seeing regressions when we start treating the tumors of these mice. And we can, in some cases, we can actually see complete regression responses. So these guys stopped the drug treatment at day 57, and you can see that there was a small fraction of, this is a mouse, for example, where the tumor looks like it disappeared, and for over 20 days, there was no evidence of tumor here. So Nori Yamaguchi is an outstanding clinical fellow in my laboratory. He's done a series of experiments with this small molecule. And what you can see, this is a PDX of a KRAS mutant tumor. This is PDX obtained in collaboration with Yelena and colleagues here. So each dot represents a tumor. This is a PDX. These tumors start about 175 millimeters cubed, and you can see they grow. And he starts treatment here, and he can start seeing regression responses. And this is, sorry, you can't see this. My apologies, but and this is a prevention study where you put them on the drug and you inject the metastatic cells, and we can see inhibition. I didn't show the data, and I should have, but Nori's also found that in some gastric models, he also sees substantial activity of this compound. And so we've sat down with Yelena and others, and she's helped us to design a clinical trial. And we're initiating a phase 1 trial. The FDA has allowed us to move forward. Andreas Sursek is leading the phase 1 here. It's at 5 institutions. So there's going to be a monotherapy dose escalation in advanced GI malignancies. And then in one group, we're going to do combination with FOLFIRI, dose escalation, and then expansion in gastric GE junction versus colorectal cancer, the 2 cancer types in which we see strongest activity. And then there's also a biomarker-driven group. And so in summary, what I want to say is we find that these gastrointestinal cancers, at least 2 of them, seem to be dependent on this creatine metabolic pathway for progression. And we also find that cancer cells can co-opt a neuronal program and to overexpress a neuronal gene and to actually release it, and that can enable them to survive very harmful metabolic stress, and that this pathway can be inhibited with an experimental therapeutic. And I think it really, we'll have to see in terms of the clinical trials whether this has, what kind of impact it has on patients. I want to thank the people who've done the work in the lab, so I'm really grateful to MSK, also for the fellows, so Dennis Xu, Ryan Moy, and Nori, whose work I mentioned has been collaborating here, and thank you very much.